So thanks, Fred, for asking me to mention this topic of some of the bottlenecks that obviously are going to need to be filled if our predictive breeding models are going to be precise enough to be of interest to breeders. And this started really in lockdown when I made a survey with several major industry players, and we all agreed that there were some very significant um, bottlenecks, really. And when you look at them, root growth and function, the role of plant growth regulators and their crosstalk, respiration, and sourcing balance, these are quite fundamental processes. And so, not surprising if we don't know enough about those in our crops, that our model precision will be compromised. Recombination frequency came up as some area that would probably open other bottlenecks, but we never really got into that. But at this point in time, we were negotiating with industry and other players, coordinated by FFR, to try and actually find funding from various agencies, depending on the level of lack of knowledge, I suppose you could say, how close to impact, how fundamental, to try and address some. So let's hope that all the money doesn't go to supporting the increasing number of wars that are breaking out in the world. So anyway, so a little bit of a tour through some of these traits. Roots are always called the hidden half. I don't need to explain to this audience their importance. And now I think that are listed on the left there. Now, of course, we have the whole area of symbiosis with the soil microbiome that needs to be taken into account. And our old-fashioned approaches of looking at roots are very labor-intensive. There's Charlotte from Salk trying to find suberized roots. And yet there are remote sensing approaches, and these have been around for a while. Marta Lopez published this back in 2010, where you can actually predict root mass between 60 and 120 centimeters using canopy temperature with an R squared of 0.88. Now, of course, that's not going to work in all soils, but even if it works in some, and we're in a major wheat production zone, that's a pretty useful and quick way of getting some information about your soil depth that doesn't involve highly labor-intensive approaches. And of course, when we move to controlled environment, the largest confounding factor is soil temperature, because in the soil there's a gradient always, and research has shown that gradient is important and will impact significantly on above ground parts. And the only way really to ensure that is to bury your phytotrons in the soil so they enjoy that profile. And that's not done enough. You could say that hormones, the other hidden half, it's almost as if we are in denial that plants communicate, and the only way they can is with hormones, as far as we know. And interesting, the Green Revolution was kicked off by a hormone-related trait, the delaproteins. But since then, there's not really been a great deal of intervention in crops with hormones, and very little understanding of how a crop will respond to a heat shock, for example. The whole area of acclimatization is not sufficiently understood that we can include it in our models. And so certainly a big knowledge gap there. There's lots of basic information, but it's putting it together and putting it into a context that makes sense in the field. And this is an example where we looked in the field for cytokinin levels in the spike, which were associated with increased grain number. And this explains other traits like fruiting efficiency, where it's a signal that's determining your grain set rather than necessarily total carbon available, total assimilates available, and probably a bit of both as we dig into that. Okay, maintenance respiration. Ironically, we know more about photosynthesis that only occurs 12 hours a day, while respiration goes on for 24. Maybe that's why, because we do have to sleep sometimes. And yet, genetic variation has been documented across and within crops. But as I say, the research is eclipsed by the promise of boosting photosynthesis so much that it really doesn't matter, I suppose. But with automated sensor technology, 24/7 research should be possible. And I just want to show this graph that shows declining yields with night temperature as a main effect. And it's very clear in wheat and in other crops it's been shown, but we don't understand really why that is. And I guess respiration is a good candidate. And work conducted years ago looking across environments with this large WAMI, we called it Wheat Association Mapping Initiative. Night temperature is a hard trait to work on because you can't manipulate it that easily. I mean, you can, but it's hard work. But if you look across environments, you can plot the function of yield response to night temperature. And when we did that, this hasn't been published, novel regions are popping up, plus you're identifying lines that can be used immediately in breeding. And we do know that night temperatures are warming faster than day temperatures, which is a bit of a worry. Plants can regulate their temperature during the day to some extent if they have water, but at night they're stuck at that temperature as far as we know. Even though recently we showed apparently futile night transpiration in our environment in the desert in northwest Mexico, which is interesting. And apparently it can be a third of all transpired water. And what's behind that? There is genetic variation. It's a bit of a mystery and something interesting to follow up on. And of course, finally, source sink balance. I'll show you the wiring diagram, which is based around that idea. Key to optimizing grain yield, implicit to input use efficiency, and is also a function of the other three bottlenecks: roots, respiration, and hormones, almost undoubtedly. And you can have a step change in yield if you can control your sourcing balance. If you say that the average cereal harvest index is 0.45 and you can boost that to a stable 0.55, that's a 22% in yield right there without any extra inputs. So pretty important to understand. And these words are worried some of those in the audience who were involved in trying to get money for a AI boosted crop stimulation project using wiring diagrams as priors. I haven't heard back yet, but we're hoping for that. So this sort of lays it out how you can use these pinnacles of knowledge that we have in other areas and to be leveraged if there was a more symmetrical knowledge. And I think that is almost my last slide, but I don't know what's the timing. But you can see if we have more knowledge on hormones, that has these knock-on effects to other areas that we perhaps know. Perhaps we will learn more about those too, but are like, for example, respiration will help us to maximize net assimilation rate if we can optimize respiration, etc. You can just take a look at that figure. I'm not going to go through the whole thing. It's a bit, I think it's fairly obvious that the kinds of benefits one gets by filling in the gaps of the jigsaw puzzle. And these are big gaps. So we may not get the whole picture until more of these gaps are filled. And finally, big data analysis retrospectively of tens of thousands of your data points and sites, hundreds of thousands actually, showing clearly that as temperature warms, more niches are being formed. So this doesn't take anything away from the need of a centralized approach for disease, proving disease resistance, or something like some of the nutritional traits that are relatively simply inherited. But as we get a more complex weather patterns, less predictable niches are likely to form. Greater niches are forming. This is what the data shows, more rank changing. And so we're going to need to be able to manage these traits, because they're going to be what determines the G by E. And if there's time, return on investment into these 4 fundamental bottlenecks is likely to deliver a high return on investment just because they haven't been investigated. So there's going to be some, I got into trouble for saying low-hanging fruit, but everything is relative. So let's say that good fruit just need the ladder to get up there, but they're there because they haven't been picked. And of course, raising the knowledge bar will enable proprietary technology to flourish, which is why we're trying to involve private interests as well to invest in this. They're not currently factored into
Matthew?
breeding.
So I think, could you start wrapping up, please? Because it's time is up.
Am I over time?
Yes.
Okay, well, I'll just stop there. I think that I think the point is made. I just say that we have the a great opportunity to explore wild relatives that have survived millions of years of climate change. So why couldn't they get us through the next century or two? But we don't. We explore very few, and yet the very few we have explored have borne fantastic returns on investment. And so there is an opportunity to find better sources of all of these traits. If we put the,
Matthew, I'm very sorry, but I think we need
I
to
have
stop.
finished actually. I was
Okay.
finishing. Thank you for
So
keeping
I
me on
think there is
time.
no time for questions unless there is something very, very short and very urgent. No? Okay, I don't see any hands up, so I would like to thank you, Matthew, for your talk.